Hey, good morning, everybody. Today we're going to be moving into Chapter 4, and as we go into Chapter 4, we're going to make our way to our next level of organization and begin to talk about tissues. So again, to recall, we kind of started off on our beginning level as we cover atoms and molecules in Chapter 2. We then moved to Chapter 3 as we focus more closely on cells, and now we're kind of going to be talking about a higher level of things as we talk about tissues, which are really just a combination of cells that have a common job or function. Now, in your book, this information can be found on page 115. On page 115, there's a really good flow chart there that allows you to start breaking down the chapters, separating it, and focusing on certain objectives. Again, I would encourage you to look at that flow chart and again, use that as a model or a template to maybe pattern your notes or pattern your studying. Also, keep in mind at the very bottom of page 115, it also leads you to where you can find a video. This career connection video gives you some more insight on why studying tissues is important. Now, we're going to talk about why studying tissues is important, and today not only are we going to go over that, but we're also going to go over how tissues are prepared, how they're fixed so we can actually see them underneath the microscope, and also talk about some special characteristics of epithelial tissue, investigating different types of epithelial tissue as well. The good thing about a lot of this information is that we've already kind of been introduced to it through lab. Again, as we continue through lab, we will continue to see tissues throughout this semester, and you'll also see the same thing in AMP2 as well. So those are things to keep in mind because these are core skills that will hopefully allow you to continue to build towards your success in understanding the body in anatomy and physiology. So as we talk about tissues, again, we want to kind of ask ourselves, why is this really important? We also want to focus on histology, which is really the study of tissues, and again, understanding why that applies in a course like AMP. We're going to talk about how the tissue is prepared, how it's stained. That's going to be really key for understanding how we actually view things in a real life setting. We won't be actually doing stains ourselves on tissues because the slides that we have already been stained, but you want to understand some of the background work that goes into preparing slides so that way you kind of understand what you're seeing when you start viewing these items underneath the microscope. We're going to talk about epithelial tissue, and we're going to discuss epithelial tissue from how it's classified and again some of its key structural components. We'll also talk about glands, which fall under epithelial tissue but specifically are about secreting topics or secreting substances, I should say, inside the body, or sometimes pushing those substances outside of the body, just depending on which type of gland they are. So again, when we talk about why does this matter, we want to understand cells because this helps us understand organs and organ systems. So again, as we talk about those structural levels of organization, we have to understand cells to be able to understand tissues, and again, we have to be able to understand tissues to be able to understand organs and organ systems. So again, these are kind of just steps along the way to make our way to the higher levels of bodily organization. Now again, when we talk about understanding histology, we'll really be able to better understand how slides are made. And again, we won't be making these, but this will allow you to see kind of some behind the scenes work that goes into it. So when you start viewing these slides in lab, you have a little bit more insight on why things appear the way they appear, why it looks the way it looks, so to say. These are things that will hopefully kind of help you better understand what you're actually viewing in a lab setting. Also, as we talk about tissues, being able to understand tissues is really critical because it helps you treat patients that might have some type of damage to their tissues or sometimes tissues disorders. So again, this is really key because many of you are going into fields such as surgical technician or dental hygiene, where you may be looking at gum tissue or you may be looking at intestinal tissues, just depending on what you're studying and focusing on. Maybe if you're an EMT, EMS individual, then you're going to be treating patients as well. So again, as you talk about a career field in some type of medical area, you're going to always be dealing with patients and always probably be dealing with cells and tissues, again, on a probably fairly regular basis, I would say. Again, it may not be an everyday thing, but it will probably be something that becomes a part of your job on a fairly regular basis. So without further ado, we're going to jump into histology. And again, if you want to talk about this or see this in your book, you can begin to see this information in your book starting on page 115. As we go into page 16, again, we start to understand that body cells are specialized, and each type of body cell helps perform specific functions that maintain homeostasis and benefit the body as a whole. So this allows us to lead up to tissues, which are basically groups of cells that have a common structure and a common function that they perform. And we're going to see that they actually are what we call the fabric of the body because they're kind of woven together, therefore allowing us to have 4 basic tissue types that we're going to talk about here throughout this chapter. So again, tissues have a common function, have a common job, and again, they're collective groups of cells. And we're going to see some examples of those primarily today, focusing on epithelial tissues. So again, histology is just the study of tissues. Anytime you see that suffix ology on the end, that lets us know that we're studying something. So again, histology is really just the study of tissues. This again is referring to kind of complements of gross, or the study of gross anatomy. And again, we're kind of studying on this microscopic level so that we can begin to learn and understand the larger macroscopic properties of tissues. We want to again understand that we have to understand tissues in order to understand organ physiology. So if we want to really understand how the body works, we have to understand cells and understand tissues. So again, as we learn and study tissues, we're able to maybe diagnose patients, maybe by something with, maybe due to something from a biopsy, as we can see in the image here. This tissue is probably taken from a patient, being preserved and probably being studied, maybe being tested for something. Of course, we may not know what that is from just this image, but again, we can see kind of the beginnings of that process being displayed. So when we talk about taking tissues from the body, we want to understand that many times we can take tissues really quickly. We also have to be sure that we're doing this safely, because when you start taking tissues out of someone's body, you want to make sure that you minimize the risk of infection or minimize the spread of disease. So we definitely have to do this in a clean and sterile environment in many cases. And again, we can sometimes do, take tissue samples from the skin, nose, and mouth. Sometimes this can be sampled with a scalpel with just a gentle scrape. Most of the time, this isn't really too invasive. Sometimes it can be, but at this point, we're kind of more at the superficial levels of the body, and we can maybe just take a skin sample or a body sample by just scraping or removing just a small, thin piece of tissue, typically not significant enough to be a deep or worrisome process. However, once we start going deeper into the body and we start getting into our solid organs or trying to get cerebral spinal fluid or blood, then of course we got to go deeper. So we may have a needle that goes in and withdraws cerebral spinal fluid or withdraws blood from the body. Again, at this point, we're becoming more invasive, and again, we have to make sure that we're using sterile instruments. We don't want to use any dirty needles or anything that's possibly contaminated. Of course, the individual would likely have on gloves, and we're just going to be taking the proper precautions. Even as I say that about gloves, I'm kind of wondering why this person doesn't technically have on gloves. Yes, they're not touching the tissues, but still, probably be better safe than sorry to make sure you protect yourself and wear a pair of gloves, of course. Now, as we go even deeper, we may even have to use endoscopic tubes. And again, we see that term, endo, that prefix that lets us know that we're dealing internally. So again, we're now going into body cavities. We may be getting to the deeper portions like the digestive tract. And again, we're now dealing with a more quote-unquote invasive level. So we get a chance to see that depending on what we're trying to study, there's different ways to collect our tissue samples. So as we go a little bit further along, we want to understand that we really are focusing on being able to provide a visual view of tissues underneath the microscope so that we can truly see this tissue and truly understand its structure and its design. Keep in mind, its structure will always relate to its function. So as we begin to look at tissues and we notice certain features, certain things that are just stand out to us, we want to understand that that will definitely link very, very closely to the function of the tissue. So those are things that we definitely want to keep in mind as well. So we have an image here that shows us kind of what a stained tissue slide looks like. And what we want to understand is that before we can view a specimen, we have to fix it to the slide, so it has to be adhered to the slide, has to be preserved, and we want to make sure that we cut it into sections or slices. Now, sometimes you may see on your slide a LS to indicate longitudinal section, indicating that it was cut along a particular plane. I'm going to let you think about which plane that was. I won't spoil that for you, but you want to think about the body's planes. We talked about the 3 different body's planes in chapter 1, so you might want to go back and revisit those, but we'll also see that in other cases a section is used to produce a cross section. So again, longitudinal sections and cross sections are really the 2 most common, and you want to try to figure out which planes are used to represent a longitudinal section and a cross section, because you'll see both of these as you look at slides in lab. Now again, you might want to ask yourself, why do we need to cut the tissues very thin? When we talk about making these cross sections or longitudinal sections, why do we want them so thin? Why do we need them to be very thin? And really the simple answer to that is that we need to be able to transmit light through the specimen. We may need to be able to transmit electrons through the specimen if we're using an electron microscope. Now in our labs, we do not have an electron microscope. Those are very expensive. You typically find those at higher level universities. But we do want to understand that you can view electron microscope images by just looking on Google or other sources, and in doing so understanding that this tissue was cut thin to allow the electrons to pass through. Same thing for light. Yes, we definitely have light microscopes, and again, we want to have that tissue thin enough so that light can actually pass through. So what we're going to begin to take a look at here as we talk about histology, let's see if I can get my PowerPoint to catch up here for me. Seems like it's kind of dragging. Okay, so we want to first talk about the fixation standpoint. And again, the tissue has to be preserved. Typically, there's some solvent used to preserve the tissue to make sure that it doesn't continue to decay as it is placed on the slide. So we don't want it to start breaking down and the cells to technically they are not going to live, they're going to die, but we don't want them to decay. If they start to decay, then they won't look like they're supposed to look, which really limits our ability to study and understand them in their true shape, form, and fashion. Again, we have to make sure that we cut it into thin slices, making sure that it is thin enough so that light can shine through them. That is very, very key and very important. If the tissue is too thick or just not thin enough, essentially, then light won't shine through, and it won't do us any good of being able to see those in or under a light microscope. One of the other very, very key steps that has to be done is staining. And this is where tissues are actually colored or dyed with some type of solution. Now, there's a lot of different staining techniques and a lot of different dyes and stains out there. So very often when you view a tissue on one image and view it on another image, it may be a different color. When you're viewing it under the microscope versus what you're seeing online, it may be a different color. It just depends on what kind of stain was used. So you can maybe think about a t-shirt. You could take a t-shirt and it can be white. You can dye it blue. You can dye it red. You can dye it pink. Whatever color you may want to dye it, depending on the dye that you use. So again, when you're talking about staining and you're viewing tissues, although the staining does enhance your ability to see, I wouldn't recommend becoming completely solely sold out on just memorizing tissues by color, because very quickly that color could change, and what you really want to focus more on are the features of the tissues themselves. Yes, the color can be helpful, but you don't want to just try to memorize based off of just color, because the colors could change and you still want to be able to recognize it. For instance, a t-shirt could be white or could be black, and it's still a t-shirt. So again, you want to just keep that in mind. Again, I'm not saying colors can't be helpful, because they definitely are, but you shouldn't put all of your trust just in the color of the stain of the tissue. So again, we see some machinery here that can be used again for cutting our thin sections and allowing us to prepare our stains appropriately. Again, being able to set the stage for studying and understanding histology. Now, we won't be doing staining like I mentioned before in this class, but when you do take microbiology, or whenever you do take microbiology, you will be given the opportunity to stain bacteria. And it's not exactly the same as staining tissues, but there are a lot of similar processes by which we prepare the bacteria, we make a thin smear of the bacteria, and we stain the bacteria, allowing us to view it very, very easily underneath the microscope. Here again, we see some of the impacts or aspects of being able to once again stain our tissues. So again, using a solution to preserve it, cutting it into thin slices, and then adding our dye to give us greater visibility and greater contrast. Now again, we get a chance to see that with light microscopy, we're using colored or fluorescent dyes, very often using colored dyes. And again, with electron microscopy, we really use heavy metal coatings, because with electron microscopes, there has to be a metal coating there for the electrons to kind of bounce off of, therefore producing our image. So again, I'm not going to be too focused on electron microscopy at this point, because that's just not how we typically operate in our class. But if you are interested in viewing electron microscope images, you can find plenty of those online. They're typically a lot more detailed and a lot more enhanced. So again, it just provides a different view of the same tissue. But again, as we study, as we learn, we're primarily going to operate on the light microscope level. So what we're going to do is watch a video here. This video kind of, as you can see, is titled From Biopsy to Microscopy. So again, how do we begin taking a tissue out of someone's body, undergo all the processes that we just saw described, and then end up with the tissue that we can study clearly underneath the microscope? That is what we're going to see explained in this video here.
This video is an overview of the steps taken in preparing tissue samples for light microscopy. This is a view down a microscope of a thin section of kidney. The kidney has been fixed, processed, sectioned, and stained to show its cellular structure. If you try to cut a piece of fresh tissue and look at it with the microscope, all that can be seen is a structureless blob. There are several obvious problems. It is not permanent, so the tissue decays. It is difficult to cut sections thin enough to allow light to pass through the section, and it lacks colour. These difficulties are overcome by fixation or preservation, provision of a support medium for the tissue, and staining. To preserve fresh tissue, it is placed in fixative for a length of time suitable for the size of the piece and the type of fixative used. For example, a specimen of this size would be fixed in formalin for approximately 24 hours and then stored in 70% alcohol. The original sample is trimmed, transferred into a labeled plastic cassette, and then into a stainless steel carrier with other labeled samples. The carrier is placed in the automatic tissue processor. In tissue processing, the water in the tissue is replaced with molten paraffin wax. To remove the water, the tissue samples go through increasing strengths of alcohol solutions. The tissue is dehydrated gradually to avoid excessive shrinkage. Paraffin wax is not soluble in alcohol, so after dehydration, tissue is exposed to a solvent that is miscible with both alcohol and paraffin wax. These solvents, for example xylol, are known as clearing agents because they also make the tissue translucent. This laboratory uses a non-toxic organic solvent, histoline. At the end of the cycle, the specimen is immersed for an hour or more in pots of molten paraffin wax. The next step is blocking the specimen in solid wax. The tissue is removed and orientated as required in a metal mold, which has been filled with molten wax. The base of the cassette with the specimen ID on its side forms a lid for the mold. The mold is topped up with extra wax and placed onto the adjacent cold plate. The wax solidifies and the paraffin block with the specimen is removed from the metal mold. Sections are cut using a rotary microtome. The tissue block is clamped into the microtome. The knife is inserted and clamped into the knife holder. The knife stage is eased towards the block. When final adjustments have been made, the stage is locked in place and the dial set at the required thickness, routinely 5 microns. By rotating the handle, the block is advanced by 5 microns and lowered down past the knife to produce a section. These sections of wax and tissue form a ribbon that can be lifted off the knife and laid down in strips. Due to compression, the tissue sections are slightly wrinkled, but most folds can be removed by floating out the ribbon or individual sections on a warm water bath. Sections are collected up onto a microscope slide and dried in a 40-degree oven overnight. The wax within the tissue sections is dissolved away by the clearing agent. The slides are then rehydrated because the clearing agents are not miscible with the dyes. This stage is simply a series of alcohol washes. The dewaxing and rehydrating steps are referred to as bringing the sections to water. The sections are now ready to be stained. Hematoxylin and eosin are routinely used to stain histological sections. Hematoxylin stains many nuclei a blue to black color. Eosin stains cytoplasm and intercellular components pink. Sections are overstained in the hematoxylin section. Overstaining with hematoxylin results in many tissue components being colored blue. The dye is then selectively removed by 1% hydrochloric acid solution until the nuclei can be seen clearly. This process is known as differentiation and is checked with a microscope. Sections are again placed in a weak ammonium solution to produce a deep blue color. The slides are then stained with eosin. Eosin stains the cytoplasm and most intercellular products, for example collagen, pink, orange and red tones. Once staining is complete, the section is a mixture of purple and pink hues. To make the section permanent, it is dehydrated in absolute alcohol and then cleared in a clearing agent that is miscible with PIX. The clearing agent makes the section translucent. PIX is a synthetic melting medium and acts as a clear glue-like substance that adheres the cover slip to the stain section. A drop of PIX is placed on the stain section and the glass cover slip is lowered onto it. The PIX will harden after the slide is placed in a 40-degree oven overnight. The mounted PIX has the same refractive index as glass. This means that in the light microscope, light rays are not further distorted as they pass through the section, then the mounted and up through the cover slip. In summary, tissue processing involves fixation, where the tissue is preserved. Processing, where water is removed and the tissue is impregnated with wax. Section cutting, where a thin section is cut from the paraffin-embedded tissue. And finally, staining, with hematoxin and eosin to give the H&E stained section.
Alright, so as you can see, there's a lot that goes into staining cells or staining tissues, right? I mean, when you go and grab those slides out of the box and you just slap them to your microscope, you don't really think about all the behind the scenes work. Now, hopefully this video didn't gross you out too much. I know for some people it might have been a little too much seeing the tissues cut and such, but it is important to understand how these are prepared. Another thing that I want to remind you of, especially as you're viewing these tissues in lab, is that when they cut tissues out of the body, very often other tissues are kind of there as well, because all these tissues are kind of connected together. So sometimes as you're viewing tissues in lab, it's going to be very important to understand which part of the slide you need to look at. It's very hard to isolate just simple squamous tissue or maybe some type of connective tissue to just be isolated. Very often you might see blood vessels nearby or other connective tissues there. Adipose tissue is something that you'll see all around. Areolar tissue is something that you might see all around. So you have to be able to distinguish between the different types of tissue, and you want to make sure that you read the slide before you put it on the microscope. You want to read the slide and know what you should be looking for, have an idea of what that looks like, and then look particularly for that area on the slide. That's very, very important and very critical, because if not, you can spend your time looking on the slide and be looking at the wrong thing the entire time. So at this point, we're now going to talk about the 4 major types of tissues, and we see those listed here. These are what I kind of refer to as the big 4, and we have some artistic views of these, but we're going to be looking at real images of these throughout chapter 4 as well. So we have epithelial tissue, which is very important for covering the body. When we talk about epithelial tissue, it covers the body in the form of skin, but also lining the body as we talk about the body's cavity. So these are really tightly packed cells that form sheets, and again, protecting the body from coverings and from lining standpoints. We also have connective tissue, which is very, very abundant. It's the most abundant type of tissue. It is there for binding and supporting the body, so we'll see it all throughout the body, and again, we'll see some examples of connective tissue as well. We have muscle tissue, which is all about movement, skeletal muscle, cardiac muscle, and smooth muscle. So only 3 that kind of fall underneath that category. And then last but not least, we have nervous tissue, which is all about control and body regulation, pretty much like what you think about when you think of the nervous system. So again, today we're going to focus primarily on epithelial tissue, but in our next discussion, we will see some more detail on connective, muscle, and nervous tissue. And again, being that we've seen some of this information in lab, hopefully some of these things start to ring a bell, and you might already be able to think of examples of connective tissue or examples of nervous tissue, and like I just stated, those 3 examples of muscle tissue as well. So again, when we talk about organs, most organs will contain all 4 tissue types, and again, this is why when tissues are cut out of the body, very often you may see multiple types of tissue on a slide. So you want to read that slide, know what you're focusing on, and be able to look specifically for that region or that area. So as we talk about epithelial tissues, again, we have an image here that kind of reminds us of the different types of tissue of the body. So we see some images that point out nervous tissue, such as the brain and spinal cord and nerves. Again, we see various areas where we locate muscle tissue and connective tissue. But at this point, we're focusing really on the epithelial tissue, which again is very, very important for protection, secretion, absorption, and filtration. Now again, like I mentioned, this lines the body's cavity. So we talked about cavities like the thoracic cavity, abdominal cavity, even cavities like the cranial cavity and vertebral cavity. These all have linings, and the linings of those cavities are made by epithelial tissue. Again, when we talk about coverings of the body, like our skin and those layers that are important for external boundaries, that is once again also made of epithelial tissue. We're going to also see that epithelial tissue is made, that glands are made of epithelial tissue. So glands like our sweat glands and our oil glands and our endocrine glands are all made of epithelial cells and epithelial tissue. So again, any secretion that you think about for the body will be related to some type of epithelial tissue, which is again very, very important. So what we're going to begin to talk about now are the 5 characteristics that distinguish or separate epithelial tissue from the other types of tissues in the body. And again, you can begin to see these in your book as you go to page 117. So again, as we talk about this, you want to be aware of these particular 5 distinguishing characteristics. And again, as we talk about these, we're going to begin to list them. The first of these is that we see polarity. And this is basically just kind of referring to epithelial cells having 2 surfaces, basically a top and a bottom. We're going to see what that is, goes into that here in just a second. We're also going to talk about the specialized contacts. And this is essentially talking about how the cells are tightly joined together. They have to be tightly joined together if they're going to form membranes or form sheets. So again, we're going to see that there's some special things that hold these epithelial cells together, giving them that tight sheet making function, which again allows them to line the body and cover the body. We're going to see that almost in every instance they will be supported by connective tissues. Typically, if you see epithelial tissue on top, you're going to see connective tissue right beneath somewhere nearby. We're also going to see that they are avascular. And again, when you see that letter A in front of a term, avascular, that means without blood flow. Again, that A indicates without, so avascular, without a vascular network, without blood flow. Now you may say, wait, epithelial tissue doesn't get blood? No, they don't. And we're going to see kind of how that is actually supported, because it's kind of rare for tissue not to get blood and still be able to live. So there's some special things that go on to ensure that epithelial tissues do actually still receive nutrients. They do have nerves. So again, there's no blood vessels, but they are innervated. So again, with the nerves, they do have a connection to the nervous system, which is again really important. Last but not least, we'll see regeneration, which as it indicates how these tissues are able to grow back. So again, we want to be able to think about those things and understand those things. And again, you can begin to see this information in your book on pages 117 and pages 118. So again, like I mentioned, as we talk about polarity, all we're basically saying at that point is all cells will have a top and a bottom. If we look at our image here, we can begin to see what we call our apical surface. That word apical, the root word there is apex, so we're talking about the top of the cell. Typically that is going to border some type of open space. As we see, it's not attached to the surrounding tissue. It's exposed to a surface or exposed to a cavity, so it's really the top part of the cell. We then have the basal surface, and when I hear that word basal, I kind of think of the word basement. So like a basement would be underneath or at the bottom of a house, so to say. This is the lower side of the cell, and this is going to face inward, and it typically is going to be attached to connective tissue underneath. So if you look at our image here, we have some epithelial cells right along our border here. Notice how these cells are tightly joined together, forming a sheet or a membrane. You can also see the nuclei in these cells, so just kind of pointing out some of those organelles that we learned about from chapter 3. But if you look close enough, you can begin to see how the cells here look different from the tissue here. So if I kind of have my imaginary line that kind of goes under here, this is what we call our basement membrane. And this basement membrane separates the epithelial tissue from the connective tissue beneath. So again, we're seeing how this polarity feature allows us to be able to notice how the cell is positioned, having its top side and its bottom side, and understanding underneath that bottom side, we're going to have connective tissue there holding it in place. So again, it's going to attach to what we call the basal lamina, and this is an adhesive, sticky sheet that holds the bottom of the cells to the underlying connective tissue. It therefore links those layers together, keeping them in place. Again, connective tissue is all about binding and supporting tissue, so we're seeing how that is involved here in regards to epithelial tissue. Again, it also helps to filter, helps with cells moving in, and it also is very important for repairing wounds as we talk about the basal lamina. So again, we'll see some close-up images of this, and again, you can read more about this in your book on page 118. So when we talk about this basal lamina, it's made of collagen fibers. Collagen fibers typically represent strength. And again, these are strengthened, fortified tissues for, again, their supporting and binding features. We also want to understand, again, that these will have specialized contacts. This allows the cells to fit closely together. Again, as you look at our image of our epithelial cells here, and these are some columnar epithelial cells, look at how tightly packed they are together. I mean, it wouldn't make sense for them to be spread out, because if they were spread out, they wouldn't really do a good job of forming a sheet or forming a membrane. So again, this allows them to be tightly packed, forming those continuous sheets, which is really, really critical for their function. Again, their structure, their design, relating specifically to their function. Again, the cells are going to be tightly bound and tightly I would say, connected together. And in order to have these special connections, again, there are going to be certain things that the cells actually possess. So when we talk about, in chapter 3, there's some things that we could kind of recall as far as tight junctions and desmosomes. These, again, are very, very important for connecting the cells and basically preventing substances from leaking through the spaces between cells. They keep the cells from pulling apart. And again, these are things that could really, I would say, create a sturdy, strong, fortified network of continuous cells, which again perform the job of having these sheets and membranes. So when we talk about these specialized contacts, again, this allows the cells to fit closer together. We primarily have tight junctions and desmosomes. Again, tight junctions pretty much keep the fluids from slipping through. So as we see, like rivets on the hull of a ship. If you've ever seen a ship before, these rivets, they are really, really important to keep the parts of the ship held together and wound together closely, because you don't want water seeping into a ship. We all know that that's one of the fastest and easiest ways to cause a ship to sink. We also have desmosomes, which holds tissues together. They're kind of like a zipper. And again, if you think about a zipper, how it brings two pieces of fabric together, well, again, we describe tissues as the living fabric of the body. So again, we see how these two specialized contact features allow the cells to once again form these really, really tight sheets, which is critical for the way that they function or behave. So here's our image of the rivets of a ship, and then here's our image of a zipper, again reminding us of the special features that are designed to hold these cells together. In addition to the first two features, we also want to understand that epithelial tissue will typically be supported by connective tissue. And again, you have these two layers that make up the basement membrane. Again, that basement membrane will typically be right underneath the basal surface of the cell, therefore supporting the cells that are resting on top. So I almost kind of think about it as like the dirt or the land that the house sits on. Again, just being underneath, giving the cells something to rest themselves upon. And again, those two layers are the basal lamina and the reticular lamina. Now again, we've seen the basal lamina earlier, but again, like I mentioned, it reinforces the epithelial layer. Again, it resists tearing, it resists stretching, and you have extracellular matrix, which is secreted by the cells in this layer, so therefore strengthening and fortifying that connection. Now beneath the basal lamina, or deep to the basal lamina, don't forget those directional terms like deep and superficial. We're once again bringing in content from chapter 1. These also consist of extracellular matrix and collagen fibers. So again, this kind of forms a soft skeleton holding the tissues together. If we zoom in on this area here, we're able to kind of blow it up and again see our basal lamina and our reticular lamina. So again, these are structures that you would always kind of be able to associate with epithelial tissues. Typically, when you look right beneath the epithelial tissue, you're going to see basal and reticular lamina. I'm not specific that you be able to identify those so much underneath the microscope, but they are important to see because it helps you begin to understand where the epithelial tissue begins and ends and where the connective tissue begins and ends. And that's going to be important because, again, when you view these slides underneath the microscope, you're going to have connective tissue and epithelial tissue on the same slide. It's very hard to just cut only the epithelial cells out of the body. It just doesn't really happen that way. So you're going to have to be able to distinguish between the two and then focus primarily on the part that you need to see, which in certain cases might be the connective tissue, in other cases it could be the epithelial tissue. We want to once again remember that when we talk about epithelial tissues, they are avascular. There is no blood supply going directly to epithelial cells. So again, when we talk about epithelial tissue, we have some of our epithelial cells here. We can see those here tightly packed together. We can see the nuclei. We even see those specialized gap junctions which are holding the cells tightly together. But notice how nerves do communicate with epithelial tissue, but capillaries don't. They're in the connective tissue and they're not directly coming in contact with the epithelial tissue. So in addition to understanding that connective tissue is there to support epithelial tissue, we also want to understand that connective tissue is there because it actually provides blood flow to eventually, well, I won't say blood flow, but basically what we'll understand is that the nutrients from the connective tissue will eventually diffuse to the epithelial tissue. So epithelial tissue is still able to get nutrients, but it is by way of the nearby connective tissue. So again, that tissue, again, being nearby, not only supports the epithelial cells, but the nutrients from this layer will diffuse into the cells in this layer, allowing them to get nutrients because they don't have a direct supply of blood. So we want to be sure that we keep that in mind. Like I mentioned, that nourishment is coming from the nearby connective tissues, because without nourishment, those cells would die. So it's kind of neat how they don't have a direct blood supply, but they are able to get nutrients. You might have noticed this yourself. Maybe you accidentally cut a piece of your skin, but you didn't really cut it deep and it didn't bleed, right? You might have had a small paper cut and you feel it. You feel it because nerves are there, so again, it is innervated, but it doesn't necessarily bleed because you didn't cut deep enough. Now, I'm not saying to cut deeper, that's not my point, but what I am saying is that if you have a very superficial cut, in some cases you may feel that cut, but it may not bleed because you did not actually break your epithelial layer. So again, just kind of give you some insight on how that actually works and is situated as well. Last but not least, regeneration is very, very important. When we talk about tissues in the body, we want to understand not every tissue in the body has the ability to grow back and regenerate easily. We're going to learn and talk more about that as we go through this chapter, but epithelial tissue, thankfully, is one that does. I mean, we've all probably had instances where we've maybe cut or burn our skin, and that skin grows back. And in most cases, it grows back really quickly. And for the most part, depending on how the injury occurs, a lot of times that skin grows back and you may never even remember that it was actually injured. Now, yes, sometimes there is scar tissue involved, but in many cases, our skin grows back and it looks just like it did before. So again, epithelial are often damaged. If you think about it, your skin is in constant contact with the outside world, always rubbing against and touching things that are hot, that are cold, that are prickly, that are sharp. So this is always going to be damaged, and thankfully, because of our regeneration features, we're able to grow it back typically in a very, very fast situation, in a very fast fashion. So when we talk about regeneration, we want to understand that we are very, very much reliant upon a process that we learned about in chapter 3. Anybody maybe want to think about something that we talked about in chapter 3 that may be important for regenerating tissues? Hopefully it's kind of coming to your mind now. Maybe you can pause and think about it. Yeah, definitely going to be mitosis. Mitosis is really key for regeneration. So again, it's stimulated when we start seeing a break in the apical and basal polarity. When those lateral contacts are broken, then there's a stimulation that says, hey, we need to regenerate more cells and undergo mitosis. Now again, we have to make sure that our cells have enough nutrition so that once again there's enough energy provided and enough nutrients provided for the cells to regenerate and grow back. And again, we see our good friend mitosis being very, very key in that regeneration process. Now, what we're going to do is talk about a clinical application, and this clinical application is going to be just referring to cancerous epithelial cells. And basically what happens in this case is that the cells really fail to respect their basement membrane boundary. So what happens is that they begin to penetrate and invade the cells beneath. As they begin to penetrate and invade the beneath cells, this spread of uncontrollable growing cells is what we refer to as cancer. Now, of course, there's different types of cancer, so we're just kind of referring to, in this case, related to epithelial cells. But notice again, here are the cells. Again, they're cancerous, so they're starting to grow uncontrollably. Then they start to break through that basement membrane. Again, we've seen that basement membrane previously. Breaks through that basement membrane and starting to invade the extracellular matrix. They may even invade deeper and move into a blood vessel. And now you start having cancerous cells that move around in the body to different places that they don't belong, and growing uncontrollably. And that's essentially what we call cancer. So when you think about cancer, one of the neat ways to think about it is really the body's inability to control mitosis. Without being able to control mitosis, we start to get larger amounts of cells, cell masses or tumors. Those cell masses begin to spread, move deeper into tissues, possibly into the blood vessels. They may start to metastasize around the body, and those are things that can definitely be dangerous and concerning, and of course, deadly. So we start to be able to see a little bit of kind of how this happens. Again, I'm not going to ask you about the ROCE activation or the locomotory protrusion. That's not the detail that I'm asking you to go into, but you want to understand that this can happen because cells grow uncontrollably and again begin to penetrate into deeper tissues. Again, like I mentioned, they can metastasize and move into our lymph or bloodstream, which means they're spreading around really, really easily throughout the body, which again is not going to be good for the individual that is experiencing this. So those are things that we definitely want to think about and just keep in mind and be able to focus on as we understand how tissues work and behave. So at this point, we've kind of built our foundation, right? We've talked about how tissues are cut and prepared and seen. We've defined histology. We began to talk about the 4 major types of tissues in general. We then investigated special properties of epithelial tissues and even talked about a clinical imbalance such as cancer. So at this point, we're now going to get into how we name and classify epithelial tissues. And again, this is going to be kind of a straightforward fashion of here's the tissue, here's what it looks like, here's where it's found, and here's what it does. And again, you want to be able to understand all of those features as we talk about the epithelial tissues. And again, you'll be seeing these hands-on in lab. I would also encourage you to find YouTube videos or other helpful sources. Again, the Pearson study area, I'd recommend using those sources to make sure you can view these tissues and see as many different images as possible. I would not recommend just relying on the one picture in your book or the one picture that you first pull up in Google. Look at maybe 10 or 12 pictures, because again, there could be different colors, there could be different things that are different, different features that are different, I should say, and you want to be able to look at multiple pictures of the same thing, so that no matter what color it is or specifically how that slide looks, you're able to really understand and interpret it because you've seen it in so many different ways. So what we want to begin with is that when we talk about epithelial tissues, they all have two names. I kind of describe it as their first name and last name. And the first name is going to always indicate the number of layers. You will either have simple or stratified epithelial tissue. Now at the top, we can see the simple indicates that we're dealing with one layer. Stratified, we're dealing with more than one layer. I won't count all the layers here, but definitely more than one. Again, one layer here. Now notice again, we see our basal surface beneath, so again that just kind of reiterates the connective tissue support that we saw previously. We also see the basal surface at the bottom and the apical surface at the top. Basal at the bottom, apical at the top. So again, reminding us of some of those features. We also see the cells are tightly packed together and joined together. So those are things that we still want to keep in mind as we're looking at these images. Now the second name is very important because it indicates the shape of the cell, and we have three key shapes. Again, squamous, indicating flat cells that are kind of squished, cuboidal, which are cube-like or box-like, and then columnar, which are tall, almost kind of like piano keys or rectangles. So again, as we talk about this, just like you have a first name and last name, epithelial tissues will always pretty much have a first and a last name applied. Now another thing I want to always remind you of as well is that when you're looking at the artistic view, drawn out images or printed images like these are going to be really nice and pretty and perfect. Once you start looking at real tissues, it may not look as easy as what you see underneath, it may not look as easy as what you see here with the artistic view. You also want to remember that the tissues and images that you see in your book, even the real images, not the artistic ones, they are the best of the best. I mean, they're in the book for a reason. They're on these medical websites for a reason. They are the best of the best. However, when you look underneath the microscope, some slides are just better than others. So you, again, will have to be sure that you adjust your lighting. Don't forget about your fine focus knob. Don't forget about your iris diaphragm. Those are going to be all really critical tools to use to view your microscope, to use your microscope appropriately and view your tissue in the best shape, form, or fashion. So, again, what you want to look for when you're looking at epithelial cells, or you maybe want to look for the nucleus, because a lot of times those will be visible. Again, they typically will be dark, stained, large structures, oftentimes purple on the inside of the cell, and we can see nuclei throughout many of the images here on the right hand side of your screen. You also want to look for the cell membrane boundaries. Like I mentioned, see if you can see the cell membrane itself and be able to see where one cell begins and one cell ends, kind of the separations between the cells. You want to look at the basement membrane, again, distinguishing between epithelial tissue and connective tissue. So those are things that you want to key in on when you're looking at this underneath the microscope. Those are very, very important things. I would say this is kind of a really good checklist to go by, and I would recommend just keeping this in mind when you're viewing tissues either on the computer or especially when you're viewing these tissues, again, underneath the microscope. So, as we continue on, we want to be sure that we understand that in some cases there's going to be multiple cell types and different layers, and you're going to have to be able to look closely to be able to know what type it is. Again, when we talk about stratified epithelial tissue, it's going to be named based off of the shape of the cells and the apical layer. So those, again, are features that you want to be able to key in on and think about. And when we talk about epithelial tissue in its two main forms, we're going to be able to see that we have the covering and lining epithelial. Again, covering the body externally like the skin, or internally, again, as we talk about lining the body cavities. But we're also going to have glandular epithelial, which is very, very important for secreting purposes. Again, salivary glands, our sebaceous glands, which are what we call our oil glands, our sweat glands. These are going to be really, really important for the body, and we want to understand that epithelial tissue makes up both sides of things. Again, the lining that we see here, and then the tubules that again might be representing the glands that we can see and indicate here. So as we talk about this covering and lining epithelial, the first of these that we're going to begin to talk about is the simple squamous epithelial tissue. Now again, you can begin to follow along with these in your book as you look on page 119. And again, simple squamous cells are flattened laterally. Excuse me. Their cytoplasm is sparse. And again, they kind of look like a tiled floor. We get a chance to see these particularly in the alveoli of the lung tissue. And when we talk about simple squamous cells, because they are a thin layer of flat cells, so again, this is our first name, one single layer, our last name squamous, so one layer of flat cells, we want to understand that these are found in areas where diffusion has to happen rapidly. Basically, we want to have things that can move across these membranes very quickly and very easily, so it pays to have a thin layer of flat cells. If we had a lot of layers of very thick cells, things will not be able to diffuse across easily. So when we talk about our breathing, which is very important for the body, of course, which of course is going to relate to our lungs, we have our air sacs there that are very, very key in transporting oxygen and CO2 across these alveolar walls. Therefore, the alveolar walls need to be thin, be able to be diffused across easily, and this is why they're made of simple squamous epithelial tissue. So we find this again in places like the kidneys, which are very, very important for filtration, and like I mentioned, also in places like the lungs where gas exchange occurs. So in this case here, we're seeing some of the thin, flat cells. Like I mentioned, not a whole lot of cytoplasm because the cells, again, are very, very thin. And in some of these cases, you can begin to see nuclei inside the actual cells themselves. Again, when we talk about the special types of simple squamous tissue, we have some that we refer to as some special types of endothelium. Again, these are named based off their location. When we first talk about the regular type of endothelium, again, it will be the inner covering. So it lines our lymphatic vessels. It lines our blood vessels. We'll learn more about blood vessels and lymphatic vessels in AMP2, but we'll see that the endothelium is really important because it reduces friction and allows the blood and lymph to flow through those blood vessels fairly easily, very smoothly, and not be restricted. We also have mesothelium, and that prefix meso kind of indicates middle. These are found in our serous membranes and also lining the body's cavity. So these may be a little bit thicker, a little bit more sturdy than endothelium, but again, very important for, again, lining our body's cavities and those membranes as I have previously mentioned. We're now switching names, so now we still have our first name being the same, simple, but now our shape has changed to cuboidal. And again, when we talk about these cuboidal cells, again, they will be cube-like. They will not be perfect squares like you would think of in a, in an artistic drawing where they're all 90-degree perfect squares. Unfortunately, it's not like that in the real human body. Now, they are cube-like, but they're just not fully cubed where they're perfect 90-degree squares. So when we talk about these, they do have nuclei that are typically spherical. We can see these spherical nuclei inside the cells here. And again, notice that these cells are making up our tubules. So when you think of a tubule, if you think of a tube, maybe you can think of the inner tube of maybe a roll of paper towels. If you take that, if you take all the towels off of that paper towel roll and you just have that inner tube, imagine looking into it like it was a telescope. And as you look into it, you're now seeing the rounded end of that tube. Well, here's a rounded end, here's a rounded end, here's a rounded end. All of these are rounded ends, and these rounded ends are made of cube-like cells. So again, these are kind of square-shaped cells. We see these here that are, again, kind of square-shaped. In some of these, you can see nuclei. But in other cases, the nuclei may not have gotten stained well. So for instance, it's not to say that these cells don't have a nucleus. They just may not have gotten stained well when the tissues were cut and stained during the process that we showed earlier. We then have simple columnar epithelial tissue, which is again going to be primarily associated with absorption and secretion. Notice how these cells have now elongated in shape, so they've gotten a lot taller. Again, still tightly joined together. You can see the nuclei present in them. So again, a lot of those same features still do apply. Again, when we talk about the apical surface, sometimes it may have special modifications in the form of microvilli and in the form of cilia. Now again, we talked about microvilli and cilia in chapter 3. So again, we're making connections to previous topics that we've discussed. And again, as we talk about microvilli, these are the finger-like projections that are typically found, or that can be found. I don't want to say typically, but because they're not always present. But in certain cells, like your digestive cells, you will have microvilli that will run along this apical surface here, or along this surface here, along the top of the cell. In other cases, you may have cilia, which again are the hair-like projections for motility. There to maybe move to move mucus or other substances throughout the body, and again, they would be along this apical surface. So in lab, you're going to be seeing some of these simple columnar epithelial cells, and in some cases, you'll see those that have cilia along the top or that may have microvilli along the top. So those are things that we want to think about and keep in mind. Again, if you want to see images of this tissue, you can find these and simple cuboidal tissue in your book on page 120. We're now going to move to pseudostratified columnar epithelial tissue, and as we talk about this one, we want to understand that this is a kind of tricky one because it appears stratified, it looks stratified, but that prefix pseudo indicates false. So you may be able to say it's falsely stratified. It looks like it's more than one layer, but it's really not. So if we look at our basement membrane right along here, which separates our connective tissue beneath from our epithelial tissue above, we again notice that we have our separation, and you want to notice that all the cells are resting on this same basement layer. So even the short cells like this one here, or the tall cells like this one here, they're all resting on that basement membrane. So even though they're different heights, they are still pretty much on the same layer, on the same level. And again, only the tallest ones reach the apical surface. This is a really good view of seeing cilia along the apical surface. So here's our top side of the cell with the cilia. Here's our basal or bottom part of the cell, again resting directly on the connective tissue there nearby. So again, the short cells that we see are unspecialized. They just give rise to the tallest cells. The tallest cells are kind of the lucky ones, and they have the cilia there to once again be on top. So again, we'll be seeing these in lab. And the trachea slide is really a good slide to look at. When you look at the trachea slide in lab, you'll be able to see cartilage. You'll be able to see pseudostratified columnar epithelial tissue. You'll probably be able to see other things such as adipose tissue as well. So the cartilage slide in lab, the trachea slide in lab is a really good slide to look at to be able to really investigate multiple tissues at one time. It is really, really a good slide to view. Again, in some cases, you might see exocrine glands there, such as goblet cells that produce mucus. We can see goblet cells here and here and here. Even in one of our previous images, as you look at some of the other types of tissue, such as our simple columnar tissue, I'll go back here just a second, you can even see goblet cells here and here. Those are pretty much the same thing. So again, in some cases, you're going to see these goblet cells that are going to be important for secreting substances like mucus that might be important for the cells and tissues involved, depending on their locations. We're now switching names, and now we're going to our stratified epithelial tissue. So we're still dealing with flat cells, but now we're dealing with multiple layers. So what I'd like for you to do is maybe see if you can count the number of layers here. Yeah, most of you are probably like, nope, I'm not doing that. And I don't blame you, because it's really pretty tough to be able to count all those layers. But I'm sure you would agree with me that it's definitely more than one layer. We can see several layers of cells here. We can also see kind of how the cells are growing here. And again, they begin to flatten out as they rise to the higher layers. And we're going to see that more specifically as we go into how our skin grows and develops, and as we move into chapter 5. Now, when we talk about stratified epithelial tissue, really and truly, most importantly, you hear these associated with protection. Again, when we think about multiple layers, anytime you add layers, you're kind of adding protection. So these are found in places where we have a lot of friction, a lot of wear and tear, where things are constantly rubbing and moving against them. For instance, things like our mouth, as we consistently eat foods or maybe place things into our mouth, a high traffic, high friction area. Again, our skin is always rubbing against and touching very many things. So we find stratified squamous epithelial tissue reserved to be in those areas. Now again, the cells further from the basement membrane, the ones up here, are technically dead in many cases, and a lot of times they have a less of a cytoplasm. They are flattened, they're kind of disintegrating. We're going to see that with the skin, that technically the outermost layer of the epidermis is dead, which is why when your skin sheds at times it doesn't hurt, right? It's hard to hurt something that is already dead. We're now going to talk about a special type of epithelial tissue, which we refer to as transitional epithelial tissue. And you can see this in your book as well as you look on page 122. Now, one of the unique things with transitional epithelial tissue is that it has the ability to stretch and return back to its normal shape. So again, we want to kind of notice that we have our basement membrane here, this basement membrane separating our epithelial layer from our connective tissue layer. Our epithelial cells in this case, again, are those transitional epithelial cells. And again, they really, really are neat. We have the basal cells that are cuboidal and columnar. So again, the cells at the bottom are more, once again, cuboidal and columnar, and the apical cells might look a little different depending on how much they're stretched. But it's really important for organs in our urinary system, such as our bladder, because our bladder has to stretch and expand, and then it once again deflates as we urinate, and then it fills back up and it stretches and expands. So if you think about it, if you urinate several times a day and your bladder is constantly undergoing this change process, and then it's doing this day upon day upon day, upon year upon year upon year, you start to understand that this is a very important tissue, right? To be able to expand and go back to its normal shape continuously over and over and over again. So again, it's a really neat type of tissue. Again, this allows the basically the bladder to undergo these different transitions or change in size, and again, it really allows the bladder to hold and store more urine when needed. So we do see this name transitional epithelial tissue used to describe the transition or the changes in shape as the bladder increases or decreases in size. At this point, we've covered really our primary types of epithelial tissue. We won't really talk about stratified cuboidal or stratified columnar because they are so rare. So they are found in the body, but they're just so rare that they won't be an important focus for us. But now we want to go back and talk about glandular epithelial. And again, these are going to be all about secretion. So again, protection, filtration, secretion, those are things that we see associated with epithelial tissues. And at this point, we're focusing more on the secretion side of things as we talk about glandular epithelial tissue. So these glands are important because they make the secretions. I mean, they are actually making the secretions that are there. The cells actually make the sweat or make the oil. And many of these secretions will have an aqueous fluid that may contain proteins. Sometimes they may contain lipids, they may contain steroids. And again, you can begin to read about this in your book on page 123. So when we talk about secretion, we want to remember that it is an active process. In our early parts of chapter 3, we talked about passive versus active processes. And again, with active processes, energy is being used, ATP is being required to generate some type of movement of a substance. Well, in this case, the substance will be whatever is being secreted by the epithelial gland. So again, we see American glands, which will secrete their products through exocytosis. And then we see holocrine glands, which will actually kind of release their entire secretory cell, releasing their secretions and their fragments. In any event, this is a very, very important active process. And like I mentioned, you can read about this in your book on page 123. Now, like I mentioned, when we talk about page 123, it tells you that notice that the term secretion can refer both to the gland's product and the process of making and releasing that product. So making and releasing the actual solution can be considered secretion, or again, actually being able to produce that secretion. I apologize there. So again, referring to the product itself, the actual secretion. So you may say that the sweat is a secretion, but actually, again, in addition to that, actually releasing the sweat will also be what we call a secretion. Hopefully that makes sense. So the sweat itself is a secretion, but the process of releasing that sweat is also what we may refer to as a secretion. So that, in this case, as we talk about glands, that term secretion could have a dual meaning. It's basically kind of what I'm referring to there. So when we talk about glands, we typically can classify them in a couple of different ways. One of these can just be based off the number of cells. For instance, if it is a unicellular gland or a multicellular gland. And again, just one cell versus multiple cells. Again, that's going to be something that you can think about as well. Unicellular glands are typically scattered within epithelial sheets. And when we talk about multicellular glands, they're typically going to be growing inward within the epithelial sheet lining and kind of going down into the connective tissue. We can also classify glands based off of where they release their products, and that's really one of the major ways in which we classify glands. Even though it's kind of the second one listed, it's probably one of the more commonly used ways by which we classify. Again, endocrine glands secreting their products inside the body, primarily hormones going into the bloodstream. Exocrine glands, their products being excreted to the surfaces of the body. Typically, when you think of an exocrine gland, we can see or feel the secretions. You can see and feel saliva, you can see and feel sweat, you can see and feel wax, you can see and feel oil. So you're starting to kind of hopefully get my drift here, being able to understand that their products are secreted to the point where we can actually touch, see, and feel them many times. We can also talk about them in regards to how they secrete their product. So again, these are different ways by which we can classify different types of glands. And again, like I mentioned, if you want to read about this more in your book, it begins on page 123, and then you can see even more on page 124. So again, here's a great example of our image. Here is a great example of the image at the top of a goblet cell. This is a unicellular exocrine gland. Again, we saw goblet cells in places like our digestive tract or in our respiratory tract for mucus production. So again, we see this single cell. We see these, once again, secretory vesicles. They are the ones containing the substance that will be released, and that substance will then be pushed out primarily through processes like exocytosis. Again, a great example of an active process. Again, the number of cells, unicellular unicellular glands. So we see some examples here as far as our goblet cell. Again, another image of the same thing. This is just an artistic view here versus a real life view of the goblet cell. So like I mentioned, these are scattered within the epithelial sheets, and we've seen those goblet cells previously. In this image, we can see a few of those there in our pseudostratified columnar epithelial tissue. We also have multicellular glands, which again can be found inward down into the epithelial sheets, going into the connective tissue layer. And again, we have some artistic images of those here. And if you want to see a few more of those, you can see these in your book on page 125 and page 126 as well. Again, as we talk about endocrine glands, we're going to talk a lot about those in AMP2 as we cover the endocrine system. So I'm not asking you to know all the details of every hormone released and secreted. We're going to save that for chapter 16 in AMP2, but you do want to know what endocrine glands are. And again, they will secrete their products directly into extracellular space. Many a times these products are going to be secreted and go right into the bloodstream. So again, hormones like insulin, there's a lot of different hormones, so I won't begin to try to name them all, and I don't want to confuse you, but hormones will typically be entered right into the bloodstream, and they'll travel in our lymph or in our blood vessels to the desired organs that they're designed to be able to target. So again, a little bit of a different nature as far as where these secretions end up, but still, again, very, very similar as far as they're both made of epithelial tissue, and they're both very important for secreting and releasing their products. Again, when we talk about exocrine glands, we're once again focusing on glands that you can see more commonly. All right, my apologies there. So again, as we talk about exocrine glands, these are going to be important for releasing products to the body surfaces or in-body cavities, and these are the ones that we're more familiar with. Again, they still release their products through exocytosis, so again, that is referring to an active process that we learned about in chapter 3, but again, they do have ducts. So endocrine glands do not have a duct, which again requires them to release their products right into the blood vessel or the lymphatic vessel that is nearby, or the surrounding tissues that are nearby. As we talk about exocrine glands, they have this duct. So here's our duct, which is basically a pathway here. And again, the products will be secreted by the gland, go out through the duct, and again, reach some type of surface or internal part of the body. So again, sweat, oil, saliva, even earwax, mucus. A lot of times with those things are kind of gross to think about, but they are again products of our very, very abundant exocrine glands, which is very necessary, very, very near and dear. So again, as we talk about gland classification, we see that they're based off of how they secrete their products as well. For instance, Merrican glands are secretory cells which just secrete. The cells themselves are not altered. They produce the secretions, and the secretions go on about their business, but the cells stay behind, and they're going to remain in that same fashion. However, as we talk about holocrine glands, they're going to secrete their products by basically causing the cells to rupture. And again, the cells, as we see here, will kind of die for their cause. The cells and the solutions are all released at the same time. So like I mentioned, if you want to read more about this in your book, you can see this in your book as you look on page 126. Again, it goes into more detail about holocrine glands and exocrine glands. And again, when we talk about holocrine glands, that prefix holo means whole or all. So again, oil glands in the skin are really the only true example of holocrine glands. Again, they're really the only example of how cells will secrete their products, how glands will secrete their products, and the cells are rupturing in that case as well. We also see some other examples versus when we talk about classifying glands. Again, multicellular glands can be classified in a number of different ways, and you can see this in your book on page 125. You have the simple glands, which are unbranched. Again, you have compound glands, which are branched. So again, we see the simple glands, which are kind of tubular here. We see the compound glands, which are more branched out, a lot of different branches here. And again, these are just kind of different in their design, in their structural standpoint. We have those tubular glands, which might be straight and have alveolar glands and sacs. And then we have the tubuloalveolar glands, which again have both gland tubes and sacs. So again, as you look in your book, you can see this information. For the most part, as we talk about classifying glands, we're going to really focus more on the endocrine versus exocrine glands. But if you want to read more about these multicellular exocrine glands, you can do so in your book on page 125. Like I mentioned, we get a chance to see more specific information. So when we talk about the tubular glands, these again have secretory cells from tubes. The alveolar glands really form small flask-like sacs, kind of in a small hollow cavity. And then the tubuloalveolar glands really have both types of secretory units. And so again, they're kind of more a little bit of the best of both worlds, so to say, having a little bit of influence from the tubular standpoint and also the alveolar standpoint as well. So we see some examples of those, for instance, like our intestinal glands as an example of a simple tubular gland. We also have gastric glands inside our stomach, which release hydrochloric acid. So a great example of those would be like what we see here, the simple branched tubular example. We have the simple alveolar, which really don't have an important example in humans. They could be found in other animals. And then we have the compound tubuloalveolar, and a great example of those is our salivary glands. So again, just kind of pointing out some examples, some of those things that you might see throughout the body. And again, understanding there can be a lot of differences in the way that glands are designed, the way that glands behave, and also where glands are actually located as well. So at this point, this covers the first half of chapter 4, again focusing on what tissues are and then specifically talking about epithelial tissues. In our next video lecture, we'll then talk about connective tissue, muscular tissue, and nervous tissue. So thank you so much for tuning in, and I hope that each and every one of you have a blessed day.